to discuss what I know about like gene cloning and gene transfer in plants. I hope this will be a useful lecture to the graduate as well as postgraduate students who want to enter into field of plant molecular biology and biotechnology. With that, we can go to the next slide. So in today's dialogue, I would like to talk about two parts. First part is cloning of genes, and the second part will be on plant transformation.
Just I'm closing my camera, है ना?
Can you go to the next slide, please? So gene cloning, like as most of you would know, it's about amplifying a specific piece of DNA and putting it in a bacterial cell, and that allows to replicate a particular gene of interest, right? And cloning vector, it's a replicating DNA molecule attached with a foreign DNA fragment which is introduced into a cell. So basically host cell, usually bacterial. And cloning vector usually has certain features which enables it to replicate by itself, and your gene of interest can be propagated in the cloning vector. So what are these features? So first is origin of replication, and then you have an antibiotic resistant gene which enables selection, and you have a cloning site in the vector where particular DNA or gene of interest is inserted. Next slide, please. So we'll be talking about PCR cloning. So certain steps in PCR cloning. So I understand that PCR topic has already been covered in this Science Dialogue series. So the steps in PCR cloning includes amplification of fragment of interest with desired restriction enzyme sites added to the primer of interest. And then you have the step called restriction digestion, where you digest your PCR product as well as your vector. So what are restriction enzymes? These are called molecular scissors. These are enzymes which precisely cut the DNA in certain sequences, right? They can recognize specific sequences in the DNA and then they can cut. So they are molecular scissors which enable the digestion of PCR product as well as your vector. And then you purify the gene of interest and then put it into a vector, right? And this step, all of you know, like it's how to amplify desired gene. So you basically design primers specific to your gene of interest, and then you also introduce the restriction sites on both the primers. So in this example given here, like we have two restriction enzymes introduced, which is BAM-H1 and EcoR1 for the forward and reverse primers respectively. And why is it so? Because you have a corresponding restriction site in the vector. So now you'll be digesting both the insert and vector with these two enzymes. So this is an example. So next slide, please. And then how do you go about insert? How about inserting the isolated fragment into the plasmid? So there are certain enzymes which enable that process. Next slide, please. And so what are plasmids? So we talked about plasmids and vectors. Plasmids are loops of DNA in bacteria, and restriction enzymes are enzymes that can cut the plasmid DNA or foreign DNA, and you basically insert foreign genes into the plasmid, and plasmid is usually a vector which is introduced into the bacteria. Next slide, please. So choosing cloning vector is really important, and vector by its nature, it can multiply inside the host, and so to replicate your DNA, once you have done the restriction fragment restriction digestion, what you do is you ligate it. So ligate it into the vector. The enzyme that enables this process is called DNA ligase, and what the end product of this cloning is called recombinant DNA. Next slide, please. So important things required for cloning genes include plasmid vectors, linker markers, which are synthetic DNA fragments containing restriction sites, and then transformation of host cells with plasmids. And then you have selectable markers that are used to confirm whether the cells have been transformed or not. So I see a hand go up there, but we'll come back to your question later once we finish. Next slide, please. Next slide. So these are the following characteristic of a cloning vector. It usually is of small size. It has something called origin of replication. It is compatible with the host organism. For example, you have specific vectors to clone in bacteria. You have specific plant-specific vectors. There are restriction sites. There are selectable markers. There is multiple cloning sites, and in certain instances they are capable of working in prokaryotic and eukaryotic system. Next slide, please. So as I said, firstly, like vector must contain origin of replication, which is recognized in the host cell. Also, it's replicated along with the DNA that it carries. Then you have multiple cloning sites, which contains unique restriction enzyme cleavage sites. And all those red arrows pointing towards the vector are the restriction enzymes or restriction sites, which is used to cut the vector at specific location. And then thirdly, like you have the selectable marker, which enables the cells containing vector to be selected or identified. Next slide, please. So here is an example of how to introduce the gene. So you basically do restriction enzyme digestion. So in this case, it's the enzyme EcoR1. And EcoR1 specifically recognizes this sequence called GAA-TTC. And you have corresponding EcoR1 sequence in your plasmid. Once the insert and plasmid is digested, you use the DNA ligase to ligate these two pieces together. So this is traditional restriction-based cloning. Can you go to the next slide, please? And once this is done, you introduce this plasmid with foreign DNA into bacteria through the process of bacterial transformation. We'll look at the steps of bacterial transformation in later few slides. But how to identify the transformed versus non-transformed cells? So you can use LAG-Z gene, like the plasmid contains something called the LAG-Z gene. LAG-Z codes for beta-galactosidase. This is an enzyme, and this actually cleaves the substrate X-gal. We'll see how it works, and it makes the colony blue in color if there is no insert. And when there is inserted in the LAG-Z site, what happens is LAG-Z site is broken and there is no enzyme encoded, and you will get white colonies, right? So all the bacteria carrying recombinant plasmid will have white colonies. Next slide, please. So that is one form of selection. I'll also talk about other ways to select the bacteria containing your gene of interest. So bacterial transformation, what you do is you add DNA into the cells, and this forms an intimate interaction with the membrane. You give a heat shock of 42 degrees to the cells. This actually stresses them and causes them to uptake DNA. And there are pores made in the cell membrane by treating them with calcium chloride. So the cells are first treated with calcium chloride to make what is called the competent cells. And these competent cells now are capable to take the gene of interest or plasmid. And that is done just by a small heat shock at 42 degrees. And following this, you need to put the cells in cold to prevent pores from closing, right? Next slide, please. So this is how the plate with blue and white colonies look like. This is a transformed and non-transformed bacteria on a agar plate. So cells are grown in presence of X-gal, and recombinants will produce white colonies while non-transformed will produce blue colonies. And the principle is complementation of beta-galactosidase gene. So LAC-Z codes for beta-galactosidase. So, and here is the reaction below, like XGAL, it is cleaved by beta galactosidase to form this blue color compound. That's how it works. Next slide, please. So there are several vectors vectors that are used for gene cloning. Most commonly, we use plasmid. There can be also lambda phage-based vectors, cosmids, bacterial artificial chromosomes, and yeast artificial chromosomes. And each of these vectors can have as large as 15 kb to 300 kb DNA. Like they can carry that size of DNA. Next slide, please. So this shown here is the easy and basic form of cloning called TA cloning, where you use the PCR product directly into the vector of insert, into the vector of interest. Excuse me, please. So shown here is the topo vector. So topo vector, as I said earlier, like it has all the features of a vector that is required, like origin of replication, the selection marker, and LAGZ. And there is this enzyme, topoisomerase, which is cleaved off when your insert comes in. Topoisomerase is removed and your gene of interest goes in. So basically what you do is you make an A overhang, and there are T sites, there are T after restriction digestion on your topo vector. So these A and T match, and you have the insert cloned into the vector. Next slide, please. Next slide, please. So the slide is not moving. So yeah. So that was the basic, easiest way of cloning. Then you have homologous recombination-based gateway cloning. So what you do here is the cloning is based on recombination. So you have a gene flanked by ATTB sites, right? And you have a donor vector which has an ATTP1 and P2 sites. And there is a gene called CCDB genes that is for selection. And when your gene gets inserted into the donor vector, the CCDB gene is removed. And CCDB gene encodes a product which is toxic to the cells. So what happens is when the gene is inserted in the CCDB site, like the bacteria can now survive. Otherwise, it cannot replicate. So, and then you have the LR reaction. So there are two reactions, BP and LR. And in LR reaction, you have the entry clone and the destination vector. Again, this is recombination-based cloning, where your gene of interest is inserted into the destination vector through a clonase enzyme, a LR clonase enzyme, and you get the expression clone. So next slide, please. And these are all examples of the donor vector, entry clone, and the destination vector. So this is a very easy cloning carried out in most of the labs these days. So you don't basically require restriction enzymes. Instead, you have this recombination-based strategy. And you can move genes between the donor vector, entry clone, and destination vector very easily. Next slide, please. And similar to that, there is something called infusion cloning. And infusion cloning, actually, you amplify a gene, and each fragment of the PCR product has a 15 base pair extension, which has complementarity to the vector ends. And then you combine this PCR product with the linearized vector in a single reaction tube with a particular enzyme. And this again works on recombination, like recombination, and then you transform this recombined vector into the bacteria. So infusion cloning method uses the 3' 5' exonuclease activity of this vaccinovirus DNA polymerase. And yeah, and then you have a similar, you can go to the next slide. So, and then you have a similar strategy called Gibson assembly, where you can put in more than one pieces of amplified DNA fragment together by mixing it up. And it's also a recombination based cloning, but here like you can have any number of pieces stitched together in a particular vector. Next slide, please. So once you have the vector with your gene of interest ready, so you can either use chemically competent cells, or you can do electroporation to introduce this gene containing vector into the bacteria. And again, as I said, you use a heat shock in case of chemically competent cells, and you use electroporation, that is a short pulse of shock for about a small 5 seconds to let the plasmid enter the bacteria by making pores. Next slide, please. And finally, like once you have the gene inserted in the bacteria, like you plate it on an agar plate containing antibiotic selection, because your plasmid has antibiotic resistant gene, and that enables the plasmid to be replicated in the bacteria, and like the bacteria containing this antibiotic resistant gene would basically survive while others would die. And then you can confirm the bacteria containing your gene of interest through either the blue white screening or through colony PCR. Colony PCR is you use specific primers for your gene of interest and do a PCR with the bacterial cells. Next slide, please. And you can also isolate this plasmid, purify the plasmid, and then send it for Sanger sequencing.
Sir, please wait for 1 minute because so many people are waiting. So just I'm allowing them. क्योंकि फुल स्क्रीन में दिख नहीं रहा है कौन कौन, लेकिन आवाज आ रहा था, है ना? One people. Okay. Please continue.
So you can use Sanger sequencing to see if your gene of interest is there. And also you can do a restriction digestion of your plasmid once the plasmid is purified with specific restriction enzymes to see if the plasmid contains your gene of interest, right? So these are 3 different ways by which you can confirm whether the plasmid containing your gene of interest has been introduced into the bacteria. Next slide, please. So we are almost done with cloning. So I can take any questions you might have. I see one hand up. So if you have a question, like please do ask, and then we'll get into the next part.
Sometimes by mistake also they raise the hand. They click. And
Okay.
if you have any question, please ask question.
So this is supposed to be a dialogue, right? So
Yes. Any question? हाँ, Bahadur ji, you have raised hand. Bahadur Ali, you can unmute.
Assalamualaikum.
Yes.
My question is, sir, कि we are using linkers at this process. What is the purpose of the linker, sir?
So
Linkers.
the linkers basically have the restriction digestion sites, right? Like, for example, like I had shown there, like you have specific restriction sites on the linker, and that linker is added to your primer, right? So when you cut with specific restriction site, you have overhangs coming out from the linker, and that can actually match with that of the sites, restriction sites in the vector. So there
I
is
request participants, those who have not gone through this cloning, I have added one lecture on different strategy for gene cloning, in which we have discussed all how to isolate the gene, 5 strategy. If gene sequence is not known, gene sequence is known, everything, and linker, adapter, TA cloning. So you can see the strategy for gene cloning on my YouTube. Since this is part of this transgenic and sir is also taking, otherwise you will not understand. But if you wish to learn in deep, so there is one video, and PCR based cloning, 1 hour lecture on PCR based cloning also available on my YouTube. So I request attendees who are new to cloning strategy, you can also visit that video. Yes, sir. Please continue.
yes, we'll go to the next part.
Yes.
So now you have cloned the gene of interest in a vector, right? Then how do you actually introduce it into plant? That's the next question. That is, how do you make a transgenic plant? So shown here is a transgenic plant expressing the enzyme luciferase. Luciferase is an enzyme that lets a firefly glow, right? You have seen, all of you must have seen fireflies. And the reason why firefly glows in the dark is this enzyme luciferase. And luciferase has been introduced in this case in a transgenic plant, and that's why it's glowing in dark. Okay, next slide. So how do you introduce a gene into the plant? Can you go to next slide, please? So one of the strategies is by direct gene transfer into the nucleus. So you have the plasmid containing the gene of interest. So what you can do is you can use a gene gun. It's also called biolistic technique. So you coat DNA with microprojectiles, which is small particles of gold or tungsten, and then you accelerate these particles using a gas accelerator to penetrate the deeper layers of tissue, plant tissue. So you can use biolistic to accelerate this DNA with the microcarrier, the particles, and that can actually lead to homologous recombination of the plasmid with the plant DNA, and then you have the gene randomly incorporated into the plant DNA. Next slide, please. And also there is this other strategy which uses agrobacterium tumefaciens. So agrobacterium tumefaciens is a bacteria, soil bacteria, and it's called nature's small genetic engineer. So why? Because this bacteria can infect plants, particularly in the wounded regions, and it can cause these wounded regions to rapidly divide, and this causes something called crown gall, right? So a rapidly dividing tissue, it looks like tumor of the plant, and this crown gall you must have all observed in the gardens. So this is because of this particular bacterium called agrobacterium. So scientists have developed clever ways to use this bacterium to introduce their gene of interest into the plant. We'll see how. Next slide, please. So something more about crown gall disease. So agrobacterium is a gram-negative bacterium, and it infects dicotyledonous plants, and it is seen worldwide. And this is the tumor that you see, the figure A shown here. All these are tumors caused by agrobacterium. Next slide, please. So some of the characteristic of agrobacterium, it's a plant parasite that causes crown gall disease. It encodes large plasmid proteins called reputed.
सर हैंग हो गया है क्या? हेलो,
हाँ, आवाज नहीं आ रही ना सर की?
नहीं नहीं,
मुझे भी नहीं आ रही है.
हैंग हो गया मैं लग रहा है.
Yes, it's back. So can
हाँ,
you?
हैंग हो गया था शायद.
Okay.
Yes. Visible?
Yes. So a portion of the TI plasmid is transferred between the bacterial and plant cells. Next slide, please. So tDNA integrates stably into the plant genome. It's a single-stranded tDNA fragment which is converted into double-stranded DNA fragment by the plant cells. And then that fragment is inserted into the plant genome. And there is 2 times 23 base pair direct repeat within the tDNA, which plays an important role in excision and integration process. Next slide, please. So what does tDNA contain? tDNA has genes which encode enzymes for auxin and cytokinin. Auxin and cytokinin are important plant growth hormones, and these hormones like auxin or cytokinin, when they are not expressed in proper proportions, when there is an imbalance, it causes tumor formation, and the cells divide rapidly and forms the tumor. So and other than genes for these two enzymes, there are genes for opine synthesis. So there is octopine and nopalin, which are carbon sources for the agrobacterium to grow, carbon and nitrogen sources for the bacteria to grow. And then there are insertion genes called virulence genes, which enables excision and integration into the plant genome. So these are the three main components of the tDNA. Next slide, please. So here is a diagram showing what is there in a tDNA or in a TI plasmid. So you have two borders, the left and right border. You have genes which are required for auxin synthesis, genes for cytokinin synthesis, opine synthesis. There is virulence genes which enable this integration process, and of course, like other bacterial plasmids, there is origin of replication. Next slide, please. So there are two different species of agrobacterium. One is Agrobacterium tumefaciens, which produces a gall, and also there is another species called Agrobacterium rhizogens, which actually infects the plant roots. Right. So we'll just focus on Agrobacterium tumefaciens today, and how it infects the plant is in presence of, when the plants are wounded, it secretes something called acetosringone. And this acetosringone in turn activates these virulence genes, which is found on the tDNA, and that enables the tDNA to be transferred to the plant. And everything between the right and left border of the tDNA is transferred. Next slide, please. So again, this is example of how the tDNA region looks like. You have right and left border, and you have genes for auxin, cytokinin, and octopine synthesis. Right? And these genes usually have eukaryotic expression signals. And again, to remind you, auxin and cytokinins enable cell division. They promote cell division. Next slide, please. And so acetosringone is a flavonoid released by the wounded plant cells, which activates Vir genes. And there are several variants of Vir genes called Vir A, B, C, D, to E, F, and G. And sometimes they can have multiple open reading frames, which can span about 30 kilobase pair of the TI plasmid. Next slide, please. So this is how TI plasmid is integrated into the chromosomal DNA of the plant after Agrobacterium tumefaciens infection. Right? So there is this recombination that happens between the tDNA and the plant chromosomal DNA. And this is the three specific regions of the TI plasmid, which is virulence genes, tDNA region containing all the oncogenes, and you have the host specificity region. Next slide, please. So you can now you know what a tDNA is. What scientists have done is they have modified tDNA to allow genes of interest to be inserted into the tDNA, and then this modified tDNA can be used to transfer gene into the plants. So in vitro modification of TI plasmid has happened. So what has been done is all the tumor causing genes are deleted or replaced with desirable genes under proper regulatory control, and certain genes are retained, like the Vir genes, right? And then selectable marker gene is added, which can help to track plant cells, whether the cells have become transgenic or not. That can be tracked by using selectable marker genes. Usually they are genes of antibiotic resistance like gentamicin or hygromycin. Next slide, please. So here on your left is TI plasmid, and on your right is engineered TI plasmid. So now you can see in the engineered TI plasmid, you have useful genes there between the left and right border. The genes for auxin and cytokine synthesis or opine synthesis have been removed, and instead of opine utilization, you have the E. coli origin of replication, so which enables this plasmid to be propagated in E. coli as well. And the virulence genes are not removed, or agrobacterium origin of replication is still intact. Next slide, please. So then the TI plasmid is introduced into the plant by co-cultivation of plant cells with agrobacterium tumefaciens containing this TI plasmid, right? So you can either co-cultivate certain plant tissues, for example, plant leaf disc, under certain hormonal conditions, flavoring what is called callus development. So callus is undifferentiated cells, which is produced by certain hormone conditions. And the figure shown here is showing transformed and non-transformed cells coming from callus, right? And then you can go back, please. Yeah, back. So, and then you can use antibacterial agents like chloramphenicol to kill agrobacterium. Once the job is done, you don't want the agrobacterium to be growing because it can destroy the plant cells. So overgrowth of bacteria can again cause necrosis and plant cells can die. So once you have infected or co-cultivated, you want to remove the agrobacterium. So use certain antibiotics like chloramphenicol, and then you use hygromycin to kill the non-transgenic plant cells. So that's the selection marker gene that you have on the tDNA plasmid. So you have genes that encode hygromycin resistance, for example, that enables the plant cells to survive on hygromycin. So all non-transgenic plant cells do not survive, or they are dead, and what is surviving is the transgenic plants. Next slide, please. So this is an example of gene construct placed between the right and left border of the tDNA. You have NPT2 genes which confers resistance for kanamycin, and you have the gene of interest, and you have, so in this case you have a Gus with intron. We'll look at what Gus is in the later slide. Next slide, please. And this is how tDNA from the TI plasmid is transferred to the chromosomes in the plant nucleus. Next slide, please. And there is this system called binary vector system where you have two plasmids. It's a two plasmid system. So it's based on the observation that tDNA does not need to be physically attached to rest of the TI plasmid. So you can have two plasmids, plasmid A with the virulence region and the host specificity region, and a plasmid B with the tDNA with your gene of interest, right? So this is called a binary vector strategy. So when they are present together, the tDNA carried by plasmid B is transferred to the plant chromosomal DNA by proteins encoded by the genes carried by plasmid A. Next slide, please. And co-integration strategy, it uses an entirely new plasmid which is based on E. coli vector, but that E. coli vector carries a small portion of the tDNA. So homology between the new molecule and TI plasmid means that if both are present in the same agrobacterium cell, the recombination can integrate E. coli plasmid into the tDNA region. So again, in co-integration strategy, you have a plasmid which can be replicated in E. coli, but that also carries the tDNA. Right? So next slide, please. And this is an example of recombinant plasmid. So you can see its selectable marker, the homologous DNA sequence, left and right border. Your target gene is inserted in between these left and right borders. You have agrobacterium tumefaciens origin of replication, E. coli origin of replication. You have a bacterial selectable marker and the Vir genes. Next slide, please. Next slide, please. And this is this is one of the examples of vector series used for plant transformation. It's called pCambia. And you see different elements in this vector. That includes a CAM35S promoter, which is a constitutive promoter to drive constitutive expression of genes. So that means the gene is expressed uniformly across all stages of plant growth and development. And you have different other things, including GUS gene, which is a selectable marker. And you can also see different restriction enzyme sites there. They are the region where your gene of interest is introduced. And CAM35S promoter drives that gene of interest. Next slide. So this is typically how agrobacterium infects the plant and introduces gene. And you have, this is how we do it. We introduce agrobacterium carrying your gene of interest to infect a plant cell, and that infected plant cell is then placed in a particular growth media to transform it into callus, and then you have several different media compositions for induction of shoot and root respectively, and then your transformed plant is selected on a antibiotic selection media, and then it's transferred to the soil finally. Next slide. So again, this all shows the different steps of plant transformation. And yeah, this is what I have explained to you before. Next slide, please. So there are certain limitations of using TI plasmid vectors. So one of them is production of phytohormones by transformed cells that prevents them from being regenerated into mature plants. And gene encoding opine synthesis is not useful to transgenic plant, and it might lower the final plant yield. So now we have actually removed these two, right? We don't have, for this specific reason, we don't have the genes encoding phytohormones or opine synthesis in the engineered plasmid. And TI plasmids are usually large, 200 to 800 kilo base pair. And TI plasmid does not replicate in E. coli, therefore it cannot be cloned in E. coli. So we have actually removed all these disadvantages or limitations by engineering a new plasmid which has new features, right? And the other limitation is transfer of tDNA, which begins from right border, does not always end at left border. Rather, the vector DNA sequences past the left border is also transferred. Next slide, please. So how do you select a transgenic plant after transformation? Next slide. So as I said, you use a selectable marker gene. So you let your cells killed that haven't uptaken DNA. So usually it encodes genes that confer resistance to a phytotoxic substance, right? So all the cells that don't contain the DNA are killed by using certain phytotoxic substance. So most common is antibiotic resistant genes like kanamycin or hygromycin. You can also use herbicide resistant genes like bielofos or glyphosate. Next slide, please. So here is an example of TI, binary TI vector of PBIN series containing the kanamycin resistant gene. Next slide. So there are also other limitations of agrobacterium plasmid. So several factors have made it easier to clone genes in dicots like tobacco, tomato, potato, peas, but it's more difficult to obtain same results with monocot. So if you remember, agrobacterium has preference for dicotyledon plants. So it's very difficult to transform monocots with agrobacterium. So in case of monocots, we basically use bialistic mediated transformation. And it's been frustrating because most monocots include wheat, barley, rice, maize. They are important crop plants. and hence most desirable targets for any genetic engineering project. And the main difficulty stems from the fact that in nature, Agrobacterium tumefaciens and rhizogens infect only dicotyledonous plants, and monocots are outside the normal host range. Next slide, please. So there are two types of. So there are different types of genes that you can actually express in plants. So you can introduce non-plant genes, which are called exogenous genes. Like you can have genes derived from pathogens, you can have genes derived from bacteria or any other organism. For example, I showed you in a plant expressing luciferase enzyme, right? So those are exogenous genes. You can also have endogenous genes, meaning genes from plants itself. So these can be genes encoding for enzymes in a particular biosynthetic pathway or natural resistant genes. Next slide, please. So this is an example of selection. So only cells that have taken up DNA can be grown on media containing the selection agent. And what you see here is a mix of transformed and non-transformed cells. So those showing the green color shoots here on your bottom figure, where you have a Petri plate and you see calyces, and those green shoots are probably transgenic shoots, right? Plants which have been transformed. Next slide, please. And transformation frequency is usually very low when you use agrobacterium mediated transformation. So its maximum is like 3% of all cells are usually transformed. So unless there is a selective advantage for a transformed cells, these cells will be overgrown by non-transformed cells. So it's usually we use a positive selection agent like antibiotic resistance. You had seen NPT2 gene on the vector. NPT2 gene codes for neomycin phosphotransferase 2, which phosphorylates kanamycin, which is a group of antibiotics, and it is commonly used for plant selection, as well as like you use kanamycin in case of bacterial selection. Next slide. So what does positive selection means? You add to the medium a toxic compounds that is antibiotic or herbicide, and then you select cells or grow them in presence of these toxic compounds, and only cells that grow in presence of these selective agent is transformed cells. You plate out and pick off off-growing colonies, and it's possible to select one colony from millions of plated plant cells it is a days of work. Actually, it's not very easy. So you require strong selection pressure, otherwise non-transformed cells can escape. Next slide. And you can also do a visual selection where you use colored or fluorescent compounds. So you can plate out about 50,000 cells per plate, and you can pick colored or fluorescent compounds, the cells expressing these colored compounds, and this way you are able to screen about more than 1 lakh cells in several days. Next slide. And other way is to use reporter gene. So reporter gene is a visual based assay. There are different reporters like GUS, which is coded by beta glucuronidase, and you have green fluorescent proteins or GFP, and you have luciferase from firefly. These are all reporter genes, example for reporter genes. Next slide. So here in this example, so this shows stable expression of GUS in a moss, transformed moss. And here you have a bombarded cell or a plant cell expressing GUS gene, and it shows transient expression. So GUS is a marker gene, so which shows that plant is transformed or not. And you have GUS gene in your plasmid, right? In your vector, plant transformation vector, you have Gus gene, and when that vector is inserted into your plant DNA, it expresses this Gus, and you can do a Gus staining, which gives a blue color. So we won't go into the details. Next slide. So again, this is transgenic plant having the gene Gus fusion, and in this case it's showing a specific root expression. So you know that this gene is expressing in the plant by looking at the Gus expression. Next slide. So this is example of cells transformed with GFP, right? So this on your left is a protoplast expressing GFP, and you have a colony derived from protoplast there, and the whole plant regenerated also is expressing GFP there. Next slide. So regeneration system, how does it work? How do you get plants back from these transformed cells? So for that, you require tissue culture techniques to regenerate the whole plants from single cells. So that itself is a big topic, and we won't be covering all of that today. So getting back a plant from a single cell is important, so that every cell has the new transformed gene or DNA in there. Next slide. So in plant tissue culture, you use different growth regulators and nutrients to regenerate plants in vitro. And shown here is regeneration of shoots from leaf protoplast. So from this plant Arabidopsis thaliana. And protoplast is nothing but the cell wall removed. So once you remove cell wall, what is left is protoplast. Next slide. So there are different transformation protocols. Most commonly used is leaf disc transformation. So you cut leaves into small discs and you co-culture it with agrobacterium. So after you do the co-cultivation, you select and you regenerate with tissue culture to get the entire plant. And you make sure that the plants have gene in every cell, right? A desired gene in every cell. In case of plants like Arabidopsis thaliana, you can use floral dip technique, which does not require tissue culture. So in this, you dip the plants, that is the flowers, into the agrobacterium containing solution. So reproductive tissue is transformed and resulting seeds are screened for drug resistant growth. Next slide, please. So we'll skip this. So this shows how leaf disc is transformed, and this shows how agrobacterium, how Arabidopsis is transformed by dipping in a solution of agrobacterium. So this technique is called floral dip. Next slide. And in recent times, you have techniques to generate non-GMO plant using genome editing approach. So CRISPR-Cas9, you must have heard, it's a popular technique used these days to engineer plants which are non-GMO. So you can make precise mutation in the plant DNA using the CRISPR-Cas system, right? So again, when you do CRISPR-Cas system, you can have different methods of transformation. You can transform protoplast, that is cells with no cell wall, using PEG-mediated transformation, PEG-mediated delivery. You can use particle bombardment to deliver the particles, gold particles containing the Cas and the CRISPR-Cas system. And you can use nanoparticle-based delivery systems. You can use meristematic microinjection, and you can transfer all types of organs to introduce your gene editing construct. For example, you can have zygotes, and you can introduce the CRISPR-Cas system using PEG-mediated cell transformation of zygote. And there is something called pollen magnetification, where you transform the pollen with your gene of interest or your CRISPR-Cas construct. And then the transformed pollen is now used for pollination to get seeds. And you can also use a de novo meristem induction system, where you use the CRISPR-Cas system in the meristematic region of the plant, and then you use the meristem region. Meristem is the rapidly dividing cells in a plant, right? Region of rapid cell division, and you can use that to grow these plants and get the seeds out of that. Next slide, please. Next slide. So again, this is using, you can actually transform, you can use ballistic approach to deliver genes into pollen grains as well, and then pollinate to get the seeds with your targeted mutation. You can get gametes, fuse them, the egg and sperm cells, you can fuse them to get zygote using electrofusion, and you can treat these zygotes and transform them with PEG mediated transfection and get a new plant out of it. Or you can isolate embryos or shoot apical meristem. You can bombard that to get using the ballistic method to get a new plant containing your edited DNA or gene of interest edited. So these are all different techniques by which you can get a new plant which is transformed, or a GMO plant. So with that, like, I'd like to end this talk. We are almost on time.